Hello, and we're back in Bio 121, and we're moving on now to the second part. What are the basic construction factors required to think about for the subcellular organelles of a eukaryotic cell? And by subcellular, I mean below the entirety of the cell. This entire structure would be a cell with all the potential modifications that could be found on one. And what we want to do is we want to think about why it's required that Mother Nature builds all of these substructures. Now, most of this is going to be sort of, hey, here's ribosome, and here's what it's doing, and this is what it looks like. And you want to start there. At least I'm hoping most of you have already experienced something like this in maybe, 5th grade science class. So I'm not teaching you anything remarkably new. It's the mechanisms later in the semester that are required that require that you understand what we're talking about now. So sort of take this material seriously, remembering that inside every different cell, there's going to be a slightly different utility for what we're about to talk about. Okay? And we should start at the plasma membrane. And the plasma membrane is made up of a phospholipid bilayer with embedded proteins in it. And you're going to see that there's an asymmetrical application of materials here. You're going to see that the glycoproteins, these are proteins or lipids. So here's a glycoprotein, a protein with some sugar moiety attached to it, or a lipid that has some sugar moiety attached to it. These structures, are going to now create that membrane barrier from the outside world. And when we say outside, we're going to say, let's go like this, outside, and let's go inside. Okay, and I want you to start drawing this inside your head that way as well, which means Mother Nature needs to figure out a way to pull water towards the cell membrane. By putting these glycoproteins outside here, it draws water to it. She doesn't need to do that on the inside. Essentially, a cell is a bag of salt water. So instead what she does is she creates those mechanisms at the other side of this phospholipid bilayer to allow for the functionality of materials inside of the cell itself. Peripheral membrane proteins are those that are found at the periphery of the membrane. Integral membrane proteins are those that pass through both sides of this phospholipid bilayer. And you should be asking yourself, how is it Mother Nature maintains this fluidity? It's going to be called the fluid mosaic model in a few minutes. So I want you to understand that she's going to take some form of a lipid structure and she's going to place it in between these to allow those phospholipid tails, which are nonpolar, to behave, literally swish like tails or seaweed in water. Okay, but notice they're facing towards one another, and these amphipathic molecules ensure that Mother Nature, even though it looks like it's a solid structure, utilizes the hydrophobic forces that are pushing, in this direction and pushing in this direction, such that you end up creating this sheet. Okay, so that's probably a lot of information, and hopefully some of it made sense to you. Okay, it's because we're going to move inward now, and you're going to see it's inside the cytoplasm where you're going to find organelles that are suspended in a gel-like cytosol. Think of like jelly and jam. You open up the container. It's not completely water, but it's, it has that semi-solid consistency due to proteins that are actually found within it. And the major nuclear structure we want to start with, of course, is the nucleolus, which has a series of responsibilities you're going to want to read about, and then it's storing around it, actually producing specific materials, tRNA, ribosomal RNA structures. But these strands here are, of course, the genetic material that's within the nuclear membrane itself. And I want you to see that the nuclear envelope isn't sealed. I want you to see that you can actually move in and out of the nucleus. It takes a certain amount of work. There's transporters that are required for that. But I also want you to see that the endoplasmic reticulum, primarily the rough endoplasmic reticulum, we should put that there, rough, R-O-U-G-H, rough endoplasmic reticulum, is here. And what this means is Mother Nature is taking materials from the nucleus, sending it through the rough endoplasmic reticulum. And remember, each one of these red dots means something. Each one of these red dots is actually a ribosome. And remember, a moment ago, I said ribosomes are really important for protein production. So that means everywhere you've got a little red dot, proteins are being made. Now, when you just think about what this could be doing inside of a cell, it means that the nucleus is probably the major regulator of protein production as it responds to information that's being sent into it so it can react. Okay, and by react I mean differentially change to keep a cell in normal homeostasis. Okay. Now, you're like, okay, whatever. The nuclear envelope, okay, separates DNA from the cytoplasm. It also separates, if you remember this stuff from middle school, transcription and translation, which is where we're going to store information and we're going to literally create the protein from that information there. And how do you actually get those materials through that perforated structure? Okay, you actually have perinuclear space here, which is between the membranes themselves. These nuclear core complexes allow materials to move back and forth such that it has a nuclear localization signal to either pass into or pass out of. Without the signal here, okay, without the NLS, it's difficult for materials to get in and out. Okay, so having said that, let's see what happens when we put the whole thing together. That nucleolus with the genetic information, notice it's unfurled here. We're now going to call that chromatin, and it's the first time we've called it something other than just generalized DNA. Chromatin is the relaxed form of DNA that is active inside of a cell. It's not running around in circles. There are things that are happening across all of these strands. But inside that nucleolus, okay, you can assemble where ribosomes are assembled from RNA and proteins. It means that along with tRNA, that this is the major production point for ribosomes. And those ribosomes are again reused for the production of proteins. What is a ribosome? There are two different sizes. There's a large subunit and a small subunit. They're used during protein synthesis. And what's going to happen is they assemble in a stepwise mechanism the amino acids that are required to be turned into a protein. Now, ultimately inside a college level class like this, we're throwing vocabulary words at you that we assume you understand. And this is a very good example of that. Previous to this moment, we assumed you understood what protein synthesis was. We assumed you understood that ribosomes are required for that. And we also assume that you understand that amino acids will be assembled into proteins. And right now, we haven't told you how any of that actually happens, but we want to make sure that you understand that it does occur, right? We'll go into the how and the why in a few minutes, well, maybe a few days. But the idea, of course, is you're going to see that you have to assemble this macromolecular machine. And that macromolecular machine has a large subunit, a small subunit. We'll have this thing that's called mRNA that came from someplace that just a moment ago we referred to as transcription, TRX. And what's happening here is the information on that mRNA is being translated. Okay. And what's going to happen is, remember that tRNA that we just saw was made inside the nucleolus, which is where that large ribosome and small ribosome are. They're bringing the specific amino acid to the specific location at the specific time, such that you can build now the proteins required for things to happen inside of a cell. Now, how does this all happen in terms of the amount of energy? You know, we're considered to be a symbiotic organism, meaning every single one of our cells, with the exception of probably mature red blood cells, have mitochondria inside of them. And this is where everybody tells me, it's the powerhouse of the cell, which is true, and I'm tired of fighting that battle with people. Instead, what I want you to see is it's the inner energy generation source, right? And what happens is Mother Nature has made two membrane-bound structures here with an inner membrane and an outer membrane. When we actually get to cell metabolism, I'm going to teach you how Mother Nature separates specific elements, primarily hydrogen protons, which is what we were talking about a couple of classes ago, so that you can generate what's called a proton motive force. And that proton motive force, because this is charged, is actually changing the location of potential energy so that you end up making the currency of your cells And the currency is adenosine triphosphate. And that adenosine triphosphate is where we store potential energy to do work inside of a cell. Now, this is really important because without these cristae, without being localized inside of the mitochondria, and this mitochondria will literally change the number of mitochondria depending upon how active you are, okay? It's a responsive unit to how much ATP you actually need. Imagine before COVID, you were going to school, running around, doing all these different things. You had required less energy, and therefore, literally inside all of your cells, fewer and fewer mitochondria would be found. Even if you tried pathetically, like me, to maintain some form of physical activity, running up and down the stairs, rowing on a machine, running around in circles, you know fine well you didn't need or you didn't make enough. This because you didn't need enough ATP to stay alive. Okay, so another organelle. These are really cool. Remember a couple of classes ago I asked you if you'd ever had a wound cleaned out by H2O2. Remember that's peroxide, hydrogen peroxide, peroxisomes. They make their own hydrogen peroxide. Which happens is they actually make materials that can detoxify, when you say poison, detoxify things that are trying to do you harm, right? They spit the material from outside here onto it, and things literally burn. I don't mean that in any weird way. I mean, is it like the reactivity of the peroxide breaks down fatty acids and amino acids so that you can actually render them less harmful to you. Okay. There are different ones. They're also thought to be part of the aging process, right? So a natural process of your body not being able to repair tissues. Your body can take things apart this way. Now, notice so far I've been speaking primarily about human cells, and the purpose, of course, is that if you're thinking about a human cell, or even if you're thinking about a tree outside your window, Mother Nature had to make sure that they were slightly different than one another, okay? Because both have these structures we're going to call microtubule organizing centers. But animal cells also have centrioles, and those centrioles are going to be important, okay? Because this is how we're going to split those cells actively. Plant cells, however, have a cell wall, and I know everybody knows this from grade school, okay? And they have chloroplasts and other specialized structures such as a vacuole, okay? And that vacuole acts more like a bladder in the sense of maintaining pressure and all of that. Animal cells don't need that because they're a sealed bag of salt water. Plants, on the other hand, need to regulate water a little bit more aggressively, okay? So here's your animal cell. I've already shown you this. You're going to see that these are the different structures that are found inside of it. Here's a plant cell, and you're going to notice the vocabulary changes differently. One thing to remember is that plant cells can actually be connected between two plant cells, and to do this, you use this thing that's called a plasmodesmata. Now, it's occurring to me while I'm sitting here, we're just jumping between cells to cells, and I'm hoping that you're asking yourself, am I following along well enough that I can define all of these? Notice there's a plasma membrane here. There's a cytoplasm. We haven't spoken about cytoskeletons yet, but there's a thing that's called chloroplast, and you're going to see it's defined for you here. And this thing is a plastid. I don't really care about those yet. Excuse me. Okay, and then you got this mitochondria. We just spoke about that. We haven't spoken about what that Golgi apparatus is doing, but there's that rough endoplasmic reticulum. Here's that nucleus. Here's some ribosomes. So hopefully if you're just doing this sort of internal check, you're getting this idea, hey, I'm beginning to get an idea of how cells are similar and how cells are different. So one of the ways that they're different is this thing that's called the centrosome. And this centrosome is made up of proteins that are aligned in a specific manner, all right? And a specific manner means those non-tubulin proteins hold the microtubules together. And a microtubule is a series of monomers of the protein tubulin. Let's go like this. Monomers of the protein tubulin that are stacked on top of each other. So even though these look like solid rods that are hollow on the inside, they're not. They're literally tubes of protein stacked on top of one another, little tiny beads. Okay? And they function. They function such that they actually have a repeated array made up of 9 triplets. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. And this actually gives you now this repeated hollow form, and that repeated hollow form has incredible strength and integrity to it. Now, I'm going to change gears in a couple of minutes, right? Because we need to think about how this is different. If we have centrosomes inside of cells, what do plant cells have instead? Well, they have an external structure to the plasma membrane. So let me make sure I go like this. They have an external structure to the plasma membrane, and the plant wall will now use this instead of peptidoglycans, okay, that prokaryotes have. They're going to use cellulose. And cellulose is an important part to your diet, but if you take a look at it, I can point to these things now and you can go, oh, I know, that's an OH group. And oh, look, there's an H group. And all these little central things here, these are all carbons. Okay, here's a functional group that was attached to it. You might not be able to tell me what that functional group is specifically, but you know now that it's attached to a carbon, and that carbon has 4 other materials attached to it. And this is the sort of thing you want to practice doing while you're reading through a textbook. Okay, so can I apply what I've learned from previous lectures to what we're actually studying now? That they assume I understand, and they understand that's why they use this information here. So before I finish, I'm going to introduce, if all eukaryotic cells have mitochondria, plant eukaryotic cells, and some prokaryotic cells too, this is where things get weird, right, have these things that are called chloroplasts. And these double membrane organelles, okay, have their own ribosomes, they have their own DNA. And one thing I didn't mention that I'm sure I'll come back to later, I focused on the energetics of mitochondria. Both chloroplasts and mitochondria have their own DNA, which means they can, that's how they respond to the changes inside of the cell. Okay, the inner membrane has an aqueous fluid that's called stroma, and a set of interconnected and stacked fluid-filled membranes that are called thylakoids. Now this is going to be important because this is the sort of stuff that is definitionally based and people will test you on, especially when we think how it is photosynthesis is going to occur. But if you take a look, here's your outer membrane, there's your inner membrane, and those inum granum stacks, which are thylakoids, okay, have a lumen, and Mother Nature is going to populate these much like she would, say, a photoreceptor cell, to collect energy and then produce intermediates that can be used for metabolism, not only for the chloroplast, but also for the cell that's holding the chloroplast, as well as the entirety of the plant. That's a pretty good place if we're going to think about pausing for a moment to stop, all right? So I'll pick up at vacuoles next, because what are they doing inside of a plant? And hopefully by the time we finish all of this, you'll be able to see how Mother Nature has tied together life on the surface of this planet.